Hi, it's Dr. Barr, and I love me some histology. We're going to talk about cell junctions. By the end of the session, I want you to be able to correlate the ultrastructure and histology of the different epithelial cell junctions with their functions and locations. In the microvilli lecture, we discussed the microanatomy of epithelial cells, and in this particular lecture, we're going to focus on structures that are located in the lateral domain. Before we start, I want you to understand the terminology and difference between cell junctions versus cell adhesion molecules. Cell junctions play a role in cell-to-cell adhesion and provide cell stability, and there are different types, the occluding type junctions, anchoring junctions, and communicating or gap junctions. Keep in mind that anchoring junctions and gap junctions are not exclusive to epithelial cells. They can be seen in muscle cells as well. Cell adhesion molecules enable cell contact as well as cell-to-cell interaction, like they're talking to one another. And there are two different types. They can be calcium-dependent molecules, such as cadherin and selectin, or calcium-independent molecules, such as ICAM and integrins. And we'll learn more about these molecules as we move forward into your lectures. Now with cell junctions, I want you to be mindful of the different types of structures here. The zonular occludins are the tight junctions. Zonular adherens help form the junctional complex along with the third molecule, which is your macula adherens. You have also your gap junctions, which are also distributed throughout the lateral domain. And the hemidesmosome is the only, again, one that's located on the basal aspect of the cell. So let's first look at the junctional complex. And it's basically located on the lateral domain near the apical aspect of the cell here that I have circled. Now on boards and exams, they can give you electron microscopy and they will ask you to identify and recognize the location ultrastructurally. So the zonular occludins are located at the most apical aspect of the cell. Then underneath there, you have your zonular adherens, and then deeper down more in the lateral arena, you have your macula adherens. Now the tight junctions are your primary intercellular diffusion barrier. They are commonly located in the small intestine and the vessels of the brain, like the blood-brain barrier. You don't want anything to cross into the brain, and so that's why they need to be tight. They are comprised of three transmembrane proteins. You have your occludin, which is located here in the red-brown. Then you have your claudins below in the green-blue. And then you have your jam, which looks like a little immunoglobulin sticking up on top there on the more apical aspect. Jams are seen with endothelial cells and mediate cell adhesion, particularly the inflammatory cells when they need to get through the endothelial cells into the deeper tissue areas. With the function of zonular occludins, we talked about the selective passage of various water molecules, but the pathway is this paracellular where it crosses the junction. And the permeability depends upon the ratio of occludins and claudins. And to compare and contrast, you have your transcellular pathway, which is going through the plasma membrane, and that requires active transport. I'm going to leave with you a mnemonic of how you can recognize the various proteins that you can see in zonular occludins, because it feels like I'm throwing a lot of terms at you. But if you can remember bear claws holding so tight, occluded jam squeezes between fingers, then you know that the tight junctions are comprised of claudin, occludin, and jam, and the paracellular diffusion squeezing between the fingers is the method of pathway movement between the cells through the tight junctions. Now the next structure we want to focus on is the zonular adherens, and you have here the molecules of interaction with the actin filaments, which are these very fine filaments inside the cell, and they provide extra resistance to force. Now between the cells you have your E-cadherin and catenin complex right here, so it forms a little lock between the cells. E-cadherin is your calcium-dependent cell adhesion molecules. One clinical correlation to understand is that if you have loss of E-cadherin or catenin expression, these cells are going to splay apart from one another, and that is one of the bases of invasive tumor. Just to kind of digress a little bit, we mentioned a few about actin microfilaments, intermediate filaments, and how do they relate to one another, right? So these are a group of network proteins that are present in the cell cytoplasm, and they have various roles such as movement, transport, cell shape change, as well as support, adherence to cell or cell to extracellular matrix, even phagocytosis. And so I want to kind of give you this kind of overview where you want to group these filaments by size. So microfilaments are the smallest, so you can see the term actin microfilaments in microvilli and stereocilia. Intermediate filaments are a little bit larger. These are your main cytoskeletal cytoskeletal molecules of the cells. So cytokeratin is seen as epithelial cells, desmin is seen in muscle cells, vimentin is seen in mesenchymal cells. Microtubules are the largest ones, and we see those in centrosomes and cilia. Now going back to the lecture, we have your macula adherens, which is also called your desmosomes. Now keep in mind they are bound to intermediate filaments inside the cell. And then you also have, again here, your cell-to-cell adherens here. And what causes these cell-to-cell adherens are your key proteins of your desmocolin and desmogliin, which cause this zipper effect to form. With hemidesmosomes, it is a variant of the anchoring junctions, kind of like what we saw with the desmosomes, but I call it a half a sandwich, because basically it's found on the basal cell. You actually have your intermediate filaments, you have your molecule here of your hemidesmosomes, and it's anchoring the cell down to the underlying basal lamina and the most underlying connective tissue here. So it provides a cell to extracellular matrix connection here, and very commonly seen in the basal cells of the epidermis. There are three molecules I want you to be mindful of, again, plectin, BP230, and urbin, which are the components of your hemidesmosomes. Last but not least, we have your gap junction, also called nexus, which are your transmembrane channels or pores, and which allow for cells to communicate with one another. They're commonly seen in bone, heart, and smooth muscle, and the channels are comprised of two halves. So a half here and a half here, a half here and a half here of connexins, which allow the molecules to pass through them, and the diameter is regulated by changes induced by calcium. So on that note, I will leave you with a recap of a table. Be mindful of the different types of cell junctions and their names, key structure, functions, and key locations. Okay, also be mindful and look at the images. That would definitely show up on exams, and especially on boards. Okay, and on that note, please contact me if you have any questions, need clarifications, and I will see you at the next lecture. Thank you. Bye.